Let's go on a journey with microbial oceanographers, scientists who study microscopic organisms in the ocean to collect phytoplankton, the photosynthetic plant-like organisms that generate oxygen for us to breathe and form the base of the marine food web. Our work starts at sea on a ship. These ships are typically 100 to 300 feet long. Most cruise ships are about 1,000 feet long. Our research vessels can typically hold 40 people. The largest cruise ships can hold close to 700 people, and they have amusement parks on them. We have board games. Scientists study all kinds of different things from these research vessels, but we study phytoplankton. How do we collect something microscopic from a ship? We use a CTD rosette, a conductivity, temperature, and depth instrument. Conductivity tells us what the salinity of the water is. This instrument has bottles called Niskin bottles in a rosette or ring around a metal frame. It is extremely heavy, so we use a winch to deploy or drop the CTD into the water. The CTD is programmed by a computer, and each of the bottles, which are open when the CTD goes into the water, can be closed at specific depths. This allows us to capture liters of water from different depths in the ocean. Sending the CTD rosette down and bringing it back up on deck can be very dangerous. Scientists and crew wear safety gear like hard hats and life jackets. The CTD rosette needs to be guided into the water with ropes to keep it from swaying. If the CTD sways too much, the cable could snap. When the bottles have all been tripped or closed at all the depths that the scientists on deck wanted, the CTD rosette is carefully brought back on deck. You can see that the CTD is swaying a bit with the movement of the ship. We always hope for calm seas, but we work in rough seas and throughout the night. Once the CTD is on deck, it's tied down. And once it's secure, it's safe for us to come over to the CTD rosette and drain the water from each of the Niskin bottles into our bottles for further study. Each Niskin bottle has collected water from a different depth, so we can capture the biology and the chemistry unique to those depths. What happens with all that water? For us, we are interested in the microscopic photosynthetic organisms, the phytoplankton, in the water. Depending on where the water was collected, there may be millions of phytoplankton in each Niskin bottle. We use a microscope to isolate individual phytoplankton and bring them back to the lab to culture them or grow them. We grow phytoplankton in a lab that has special incubators. These incubators look a lot like refrigerators, except the lights in these don't turn off when you close the door. Instead, they maintain a constant temperature and light level. Phytoplankton are photosynthetic, so they need lots of light. And now I'd like to introduce you to one of the phytoplankton species we have growing in the lab right now, in a segment I'm calling Better Know a Marine Microbe. Amelia huxleyi is a coccolithophore. It's photosynthetic, it's very small, about 1/10 diameter of a single human hair, and it's covered in calcium carbonate plates called coccoliths. Plumes or large patches of these coccolithophores can be seen from space.